Whole genome sequencing allows us to read the DNA sequence of an entire genome, including both the genes and all the other DNA. But how do we get from a patient sample to a genomic report that can help inform patient care? Let's start with the sample. A variety of sample types can be used to collect DNA for sequencing, depending on the clinical context. For example, blood samples are often used for rare disease, tumor samples for solid cancer, blood or bone marrow samples for hematological cancer, and samples from the infection site for infectious disease. When the sample is received by the laboratory, technicians need to extract the DNA in preparation for sequencing. The DNA is released from the cells, purified, suspended in a liquid, and then either stored until needed or prepared for sequencing. Technicians then perform a series of steps called library preparation to get the DNA ready for sequencing. This includes breaking the DNA up into fragments as short as 150 chemical bases, represented by the letters A, T, C, and G, or as long as 100,000, depending on the technology used. Then, the sample is loaded into a sequencing machine, which reads the order of the bases in each fragment. The DNA sequence of each fragment is called a read. The sequencing machine outputs vast quantities of data, typically thousands of reads stored as text files, which need to be processed. To do this, bioinformaticians run the data through software programs. This is known as a bioinformatics pipeline. First, the reads are mapped back to their original position in the genome based on a known reference sequence. This is called alignment. The goal is to piece together the reads to rebuild the whole genome sequence of the original sample. Then, this sequence is compared with the reference genome to identify any differences, known as variants. This is called variant calling. Software is then used to predict the functional effects of any identified variants. This is called variant annotation. Once the variants have been identified and their likely effects predicted, the results need to be clinically interpreted to determine the significance of those variants. This can be challenging and may involve discussion between scientists and clinicians. The results are then presented in a genomic report for the clinician to discuss with their patient, completing the whole genome sequencing journey.